Thank you for having me. This is a perfect connection to have a perfect platform to present this information on, and I look forward to just kind of expanding our capacity to know about sickle cell and how to approach it, what it is, and some of the evolutionary process of understanding it. I first want to start off um, by giving thanks because we could all be different places right now, but we chose to be where we are right now. So I know each of us has a special connection within the sickle cell community that the information that we share can go out and we can all be ambassadors of the information as we work towards shifting the paradigm within the sickle cell community towards more of a healthy balance. So I want to give thanks, first of all, to our creator for giving us a problem to solve because I personally believe that we get problems and solutions at the same time. And, it's not always the problem, right? It's just kind of like, it's not the problem that's the problem. Sometimes it's just how we understand the problem. Um, that's the problem. So if we can share my screen, what I want to do is I want to go into a quick video by Dr. Fatima Jackson, who is a professor of biology at Howard University, and is also the director of Cobb Laboratory of Research. So I think in regards to sickle cell, um, with regards to how we approach sickle cell at Creation of Society, it's an information platform that works towards educating the public on the evolution of sickle cell using art, entrepreneurship, and agriculture. And so a lot of the things that we do is apply research and apply sociology so that we can bring more information and broaden the scope as in regards to how we understand, treat, and approach sickle cell. So once I get my screen shared, what I want to do is I want to play a 5 minute, started off with a 5 minute video of some research that was brought to us by Fatima Jackson. And it talks about the co-evolution between plants, people, and parasites.
You should be able to share now.
Okay.
I say this is only 200 miles across here, and probably about 50 miles, 75 area, the fall Cipro malaria. And I began looking that there were significant differences in the primary genetic trait that we thought at the time controlled response to malaria, fall Cipro malaria. And I began looking at that, going back to the literature. And at that time, colleagues at Rockefeller and Albert Einstein were looking at cyanate salts as with their urea base as a way to inhibit sickling. And I thought, wow, that's really interesting. I wonder where I could get 0.3 molar cyanate. And then I realized from plants, from cassava. So in this pattern in Liberia, what we saw was that in the southeastern part of the country, down here near Cote d'Ivoire, very low frequencies of sickle cell. But in the northwestern part of the country, high frequencies of sickle cell. Remember, I said this is only 200 miles across here, and probably about 50 miles, 75 miles deep. So very small country, not a lot of cultural diversity, and yet significant differences in the frequency of sickle cell. And then I realized that in the northwestern part of the country, people were eating cassava, but only as a green vegetable accompaniment to rice. And in the southeastern part, they were eating cassava every day. It was a major staple. So their exposure to the plant phytochemicals was much greater in the southeastern part of the country than in the northwestern part. And so I just kept thinking about this. Well, so how would this influence the frequency of sickle cell? And how might the diet kind of knock the legs out from under the frequency of a disorder like sickle cell. Because as we know in genetics, there's no free lunch. You know, you get a gene that looks adaptive in one setting, but it usually also has some deleterious aspects. What the researchers at Albert Einstein and Rockefeller had found out was that if you exposed red blood cells to cyanide, they were giving it orally, or cyanate salts, or urea-based compounds, that you would get something called carbamylation. And what this did was it simply modified the protein such that the red blood cell had a longer lifespan, it held onto the oxygen better, and it was more resilient against deformation as it passed through the capillaries. So this was all very good for people who had sickle cell anemia. It meant that their red blood cells would behave more like normal red blood cells. Of course, the downside was that the metabolism of the red blood cell is very similar to the metabolism of the lens in the eye. So these people were becoming blind as they were also being treated for sickle cell anemia. And I thought about this and thought, well, how do we establish that balance between poison and non-poison? How do you manipulate the diet so that you get enough of the toxin to interfere with an unfortunate and deleterious metabolic event, but not enough poison to kill the person or produce a whole nother set of problems? So here's the cassava root. It's tapioca. If you say tapioca, you would hardly recognize that this was the root that it came from. It's a tuberous crop. It's pan-tropical, which means that it's grown throughout the tropics. And when I came back from doing my dissertation work and I started working at Berkeley, and Berkeley, as you probably know, they always want to know, what have you published? Where is it? Give it to me, show it to me. So this whole publish or perish business. So I figured if I worked on cassava, I couldn't lose because it's full of cyanide. So I'll either prove it's good for you or I'll prove it's bad for you. It was a calculated risk. And so when I would do measurements of the residual cyanide, even in the cooked product, there was still quite a bit. But what really got my goat was driving back from Monrovia to the small town where we were living in Liberia at the time, and seeing a woman walk up onto the highway, peeling a cassava root and eating it raw. And I thought, my God, what is she doing? She was eating it raw. So the idea that people would consume this root without processing it, other than peeling it, was just amazing to me. You can also eat the leaves. These are the leaves, not to be confused with marijuana leaves. But these leaves also contain these cyanogenic glycosides. Now, this is a part of a family of glycosides, and you're probably familiar with the glycoside amygdalin from almonds or apple seeds, or, you know, they always tell you don't eat the cherry seeds. Eat the cherry, but leave the seeds alone. Spit out the cherry, spit out the apple seeds. And cyanide is ubiquitous in nature. It's so common in plants that we feel that there's probably multiple evolutionary benefits for having cyanide. And this is the cyanogenic glycoside. And as Mark alluded to, you have the enzyme on one side of the cell wall, the substrate on the other side. When you pull the root out of the ground, or when you peel it, or when you macerate the leaves, then you put the enzyme and the substrate both in contact with each other. Voila, cyanide. In fact, in the markets in Nigeria, they'll take the leaves, pass it through a meat grinder, and you can actually smell the cyanide as it comes off as the leaves are crushed. So this is what it looks like. It's pan-tropical, initially in South America. Then the Portuguese pick it up in the 1600s and bring it to Africa. And the people do what they always do with roots. peel it and boil it. And it's a Native American cultivar. I think that it was probably initially cultivated about 6,000 or 7,000 years ago. When we talk about coevolution, what we're talking about is reciprocal genetic influences by non-overlapping gene pools. So for example, we eat a tomato. The tomato and us, we don't share the same gene pool. Maybe metaphorically, but in actuality, there's the tomato genome and then there's the human genome. But the products of the tomato can influence the functioning of our genomes, and our actions can influence the functioning of the tomato genome. So that's what we call coevolution, where the evolution of two non-overlapping organisms, one influencing the other, they will merge together. They will mutually influence each other. And usually we talk about these coevolutionary dyads, that is where two different organisms come together. But something's really interesting about that, because when you have a successful dyad, it usually attracts a third species. Kind of like how attractive married men are to single women. I don't know, maybe it's something that married men give off. And then single women say, well, the joke is that a guy shows up with a really attractive woman and the men say, I want a woman like that. A woman shows up with a really attractive guy and the women say, I want that guy. So there's this tendency for these coevolutionary dyads to attract a third species. And that's what I think was going on in Liberia. And I've tried to very crudely show this here in this diagram. That is that the diet that the people consume has an adverse impact on both the sickle cell. Well, it's not adverse. It's actually a positive impact on the sickle cell by preventing sickling. But that same carbamylation has an adverse impact on the on the malarial parasite, because proteins of the malarial parasite will also become carbamylated and be less functional. And then of course the sickle cell, if it's not totally sickle cell anemia, but sickle cell trait, then it's beneficial in terms of protecting against malaria. And there's another enzyme, glucose 6-phosphate dehydrogenase, which is just a minor enzyme in the hexose monophosphate shunt. So as you're driving down the glycolytic pathway, you turn left and there's the hexose monophosphate shunt. And that G6PD helps to protect the integrity of the cell membrane, the red blood cell membrane. So it looks as though the diet can also decrease the active form of this enzyme, which also seems to protect against malaria. So this is why we have this pyramid-like triangular figure here, where the diet is influencing not only the human and the metabolic processes of the human, but also influencing the parasite, in this case, Plasmodium falciparum, that is interested in consuming the same metabolic products from the human. Because the parasite wants the contents of our red blood cell for its own growth and development. But now we're altering their access to our red blood cells through the diet. That's simply human plant parasite coevolution, where the evolutionary trajectories of all three species are being modified. They're modifying each other, but it's like a symphony all coming together. So on top of that now, we have the epigenomics. These are the chemicals that surround the genome, and these chemicals can influence gene expression. They don't change the gene, but they change gene expression. And it's so fascinating because it's a It's inherited. That is, you are carrying the epigenetic markers of your grandparents, which is kind of scary. It means that what they were exposed to has an influence on how your genes express themselves. Just before the talk, I was speaking with one of our colleagues here about this recent article that came out. I believe it was published in PLOS, but it was reported through Sigma Xi, that fear has been shown in mice to be transmitted intergenerationally. Fear, mice. Are you kidding me? This is just amazing. So what else? You know, and it looks as though our susceptibility to diseases transmitted intergenerationally. These are not genetic changes. These are changes in the chemical tags that in proximity to the genes can influence gene expression patterns. What a great source of chemical tags dietary phytochemicals are. So what we eat can influence the tags that influence gene expression patterns. So even when we don't see changes in the genome, we can see changes in gene expression. And gene expression is the foundation for the phenotype. And the phenotype is the direct object of natural selection and evolution. You know, sometimes we've become so genocentric that we think everything is reducible to the gene, but the epigenome, I think you'll show, we'll see over the next decade, is increasingly important in explaining differences, the differences that we see among an otherwise fairly homogeneous species, our species, because we're young and we're evolutionarily young, and we have not diverged from each other that long. I mean, all of the differences that we make such a big deal about are so superficial, almost to be laughed at. So that when our extraterrestrial colleagues come from outer space, they'll have a difficult time differentiating us from chimpanzees, much less into all of our little social categories that we have, you know. So that gets us to this question of co-adaptation and, you know, how deep are the links between the biology of plants and the biology of people. And I would posit that the links are exceedingly deep, because as we have modified the biology and behavior of the plants that we consume, they have modified us. They call to us with their chemicals, or they push us away with their chemicals. They distinguish between friend and foe, insider and outsider. How many of you have been to Jamaica and had that ackee fruit? The ackee fruit, if you don't consume it at the right time, it will kill you. It will put you in a coma. What a great way to distinguish between native and non-natives. You know, apparently we have a difficult time doing that. Wasn't there a passage, and there's probably some biblical scholars here, wasn't under adverse conditions like war? But plants and response to plants kind of gives us that insight. It's amazing. So our behavior, our biology, our metabolism is very much skewed by the foods that we eat. And because we continue to manipulate these foods like corn and rice and wheat, including now the intentional genetic modification of those plants, we can expect that we're going to see some differences. We're going to see some changes, especially when all of those modifications in the plant abut against human biodiversity. In fact, it's kind of an unknown process. How will people respond to genetically modified corn? Now, we've always been modifying corn. I mean, corn today is more like a candy bar than it is a grain. It's so full of sugars and so little protein compared to the ancestral corn. But all of this is changing us, and I know we talk about it being a brave new world, and in a sense it is, but it's a brave new world. Much of it is of our own making and our own responses to the manipulation of other life forms on this planet. So I want to end with that and open it up for some questions. So I'm sure I've provoked quite a few.
Can everybody hear me? Okay, so yeah, I think the creator for Dr. Fatima Jackson, she's put so much into perspective, and then we're going to build on that through this presentation about this relationship between the foods that we eat and its ability to have an anti-sickling impact on our bodies, on our blood, and how this can be very essential nutritional planning for the sickle cell community. So from here, I want to go into my PowerPoint. Are you able to see the PowerPoint? No? Okay. New share. Here. Share. Can you see it now? Okay. So the goal of this presentation is to broaden the scope of how we understand the genetic phenomenon known as sickle cell, who it impacts, why does it exist, and how the efficacy of Moringa aloe can bring the disease under control in a natural and non-invasive way. So that last part is very important when we think about where we are in the world, and for the most part, who's in control of um, the specification or the classification of our genetics and what process that we will be going through as a way to either heal or bring under control some of our genetic expressions, right? So we want to talk about how we can bring it under control in a natural way using food. So Creation of Society is an information platform that educates the public on the evolution of sickle cell using art, entrepreneurship, and agriculture. Our vision is to foster dialogue and action that revolves around community operated schools and a family unified school district. Our mission is to create pathways that encourage learning, imagination, and the creation of societies that embrace health, wealth, wisdom, and justice. Our strategy is to practice analyzing the language that revolves around sickle cell and correct the narrative as it relates to the genetic upgrade that defends the body from malaria. This foundation will allow us to implement our mission and vision simultaneously. So what got myself into this work was my son, who was diagnosed with sickle cell anemia at birth, and going through the hospitals, I was able to see how polarizing some of the medical care for our children who had sickle cell were, especially when I consider the invasive techniques for children that are 1 years old or 2 years old, and those processes, how it wasn't a gentle process. And a lot of the times we don't really hear about um, our children having sickle cell up until maybe the first 4 to 6 months, once the fetal hemoglobin is fully out of the child's body. And so from that point, we kind of go into a new space where we haven't had a real understanding of what the blood transfusions are looking like for these young children, or what the process is. And so going through that process with our child, when he was just about 1 years old, it kind of forced us into doing more research into how we can address sickle cell using nutrition, so that we could more or less offset some of the handling within Western medicine, as it in regards to how they approach sickle cell. So our foundation of understanding it is nutrition. So what is sickle cell? It's an evolutionary reaction to malaria from mosquitoes spreading infected blood from victims. When it reached certain people, their blood cells sickled to minimize oxygen to the parasites within the bloodstream while neutralizing their ability to reproduce. So in World War I, malaria was actually considered to be the third army. So when you have groups of troops entering into areas in the world that they are not native to, what's happening is that when they set up their tents, um, and they're out and they're moving around or eating, they would get bit by a mosquito, and that mosquito would influence the health of the body with this malaria parasite entering into it, and you would have thousands, if not hundreds of thousands of people either being sidelined, not able to fight in the war at all, or even succumbing and dying from malaria. So it was considered to be the third army for invading troops that were coming into regions that didn't belong to them, but they were trying to control an area. They would be fighting against the people, and then also having to fight against the mosquito population um, that would be essentially neutralizing their ability to fight the war altogether. So, sickle cell is a genetic survival trait, and the lifespan of this trait has experienced a disastrous disruption by many factors, considering environment, industry, and its effect on food choice and cultivation. So, when we consider how much the environment has changed, um, when I think about our environment, when I think about some of the things that our Black farming community has been faced with for the last few centuries, as far as land, access to land, and their ability to grow food in such a way that they can treat some of the health elements in their community or within their families from food, being able to stabilize their nutrition through growing food. And then industry. So the development of our industry, and how it's very necessary for the industries that we have now to have a lot of concrete. And so a lot of this concrete is going over areas that used to be a lot of fertile land, um, that could have been land that food could have been developed on. And so the industry and the environmental shifts, some of them being a byproduct of colonialism, have shifted our ability to cultivate food and has disrupted our food choices altogether. And so, what does this do? Is it produces certain diseases now that we weren't necessarily familiar with as far as understanding them as a disease, but because of the foods that we're eating are at a standard that is um, alien to us, then we are developing these different um, negative genetic expressions. So according to the CDC, sickle cell disease affects millions of people throughout the world and is particularly common among those whose ancestors come from sub-Saharan Africa. Sickle cell disease affects approximately 100,000 Americans. Sickle cell disease occurs among about 1 out of every 365 Black or African American births. Sickle cell disease occurs among about 1 out of every 16,300 Hispanic births. About 1 in 13 Black or African babies is born with the sickle cell trait. So we can understand that here in the Western world, sickle cell anemia is more dominant in our community than it is in any other communities here in the Western world. So understanding some of the sickle cell nutrients, and this is some of the work that Dr. Fatima Jackson was working on as well when she was discussing some of the coevolution between plants, parasites, and people. So the current ratio of sickle cell anemia to sickle cell trait is 1 in 50 for African Americans and less than 1 in 1,000 for tropical Africans. This etological disparity is attributed to an anti-sickling agent, thiocyanate, found abundantly in staple African foods. American staple foods have negligible sickle cell nutrients. That means that the foods that we have here in America does not feed us on a molecular level, which creates a capacity for us to have molecular diseases because we're eating in such a way that we may feel full physically, but we haven't fed ourselves. We haven't fed our cells. We fed ourselves, but we haven't fed our cells. And our cells also need nutrients so they can operate effectively and efficiently. But when we look at those numbers, so sickle cell anemia to sickle cell trait, and that's 1 in 50 for African Americans. And the tropical Africans is 1 in 1,000. So we have to be able to understand how great the impact of food is on our health, right? And also our genetic expression. So land sovereignty. This is a huge conversation happening right now all around the world. But this one is unique to ourselves and some of the challenges that we have to overcome. So foods high in thiocyanate continue to prevent the destruction of red blood cells. And people on the continent of Africa and in the Caribbean who have regained their nutritional staple foods experience less sickle cell anemia while still carrying the trait that prevents malaria. So if we look at a couple of these concepts, nutritional staple foods. So the idea of having a staple food is basically like a food that someone will eat every single day. Depending on what area you're growing up in and what's your social economic position, your staple foods may be different. You may have people who eat a lot of fast food, and fast food has become their staple food. You may have people who eat a lot of meat, and the meat has become their staple food. You may have people who eat a lot of beans and rice, and beans and rice has become their staple food. So staple foods is basically foods that you consume a few times a week or even daily that begin to produce a certain effect in your body, right? So when we look at the concept of sickle cell anemia, we have to kind of break that word up a little bit. So we understand sickle cell as a genetic adaptation to malaria, but anemia essentially is a nutritional deficiency. And so a lot of the times when we think about anemia, the coding that's in anemia is letting us know that there's not enough iron in our food, right? Which is producing anemic. It's not enough oxygen. It's not enough iron. And oxygen and iron, they work very much hand in hand inside the hemoglobin. The oxygen is actually what's supposed to be fortifying the iron should be fortifying the cells so that the cells can carry oxygen throughout the body. So when that's not happening, it produces an anemic result, right? Which is saying that what should be actually a staple food for people who have sickle cell anemia are foods that are high in iron. And what that would do is it could kind of balance some of that disease. So survival and evolution. So potassium thiocyanate binds through a process called carbonylation to the site of error on the sickle cell hemoglobin molecule inside the red blood cell and corrects it. The shape and lifespan of the red blood cell are normalized by this reaction. So I chose to talk about potassium thiocyanate because it's actually a product that we carry through our information platform, and it's been developed specifically for the sickle cell community to have an anti-sickling impact on the blood. So we're not just going to talk about the problem without offering solutions. So moringa is a nutrient-dense food that originates in India, and moringa has long been used in traditional medicines and cosmetics needs throughout ancient Africa. And so moringa being one of the most nutrient-dense foods, this is also a product that we offer on our information platform as well. And so our goal is to be able to have this particular product, moringa, because it's something that we can cultivate here in California, be available to the sickle cell community at no cost. So our goal is that they can come in, we can give them a special promo code, and they'll be able to get it on a monthly basis as a way that uh, they can create a staple food out of the moringa. The healing potential in the leaves and seeds of this tree, when we're talking about moringa, will have a positive effect on children overcoming sickle cell because of the iron richness available, as well as the thiocyanate within its molecular structure. So not only does the moringa have the capacity to be rich in iron, it also has this anti-sickling chemical thiocyanate in it as well. And so a lot of the times when we hear about children who have sickle cell, the narrative is that they're in and out of the hospital and they're having all these issues, they're having these, monthly transfusions and they're always sick. Um, and that was the case for my family or for our son, maybe for the first few years of his life. Once we discovered the isothiocyanate as well as the moringa, um, we started using it as a staple food. And our son, he's 10 years old now, and in the last 6 years, he hasn't been hospitalized at all. And we have to um, intentionally understand and contribute that to his nutrition, with him being a vegetarian and also him using the products that we're making available to the sickle cell community on a daily basis. What it has done is it's created a inner harmony inside of his body so that he can stay healthy. And it's not that he doesn't, occasionally get sick. It's just those sicknesses, or when he does get sick, is something that we can manage, opposed to having to um, take him to the hospital because it's out of our hands or we don't know what to do. Um, and so I think a lot of that, well, I think a great majority of that is, the environment, of course, and then also the foods that he's consuming on a daily. Okay. So sickle cell and epigenetics. Dr. Oji Agbai proposed viewing sickle cell as a thiocyanate deficiency anemia, affecting people only if they are homogeneous for sickle cell trait rather than a genetically determined disease. So this is a very important concept here, very revolutionary within the sickle cell community to be able to understand this as something different than a blood disease, but to propose of understanding it as a thiocyanate deficiency, which means if the mother isn't consuming these kind of foods and she's carrying a child who has the potential to have sickle cell, the child will be born without this molecule in its blood, or the family doesn't have knowledge of it, and so the child is born, and the only approach to understanding it from a Western lens is saying that it's a blood disease. But one thing about how Western medicine has been developed, that there hasn't been uh, a lot of time or energy being placed into nutrition, right? So the concept or the capacity to be understood as thiocyanate deficiency is very small unless you step outside the scope of Western medicine to be able to understand ourselves from more of a natural lens. And so I think that this is actually one of the ways that we can move forward into a better future for the sickle cell community is for us to be able to understand sickle cell anemia as a thiocyanate deficiency or a nutritional deficiency that actually points us into a better direction about how we can resolve some of these isms associated with it. That it can be resolved through food and it can be resolved through understanding that thiocyanate has to be used daily because this is a molecule that we're not getting at the capacity that we need through the standard American diet. So this example of epigenetic factors that affect how our genes may or may not cause disease or other visible attributes. People who are homogenous for the sickle cell trait and still don't get the disease because they eat cassava have a genotype but not the phenotype. So the phenotype is saying that the expression of the sickness or the body aches or the sickle cell crises, those are the things that are the genetic expression, right? But we have to get to the point where we can understand that, okay, if somebody has sickle cell, so for me, I have sickle cell, but I don't have sickle cell anemia or the constant sickling of my blood cells. So I may have the genotype, which means that sickle cell is something that is in my genetics, but I don't have the phenotype, which is the expression of these constant crises and things like that. But through the food, we can produce that within the sickle cell community, whether you have sickle cell trait or if you have sickle cell anemia, we can work towards suppressing the negative phenotype through using nutrition. And one of the things that we advocate for is the thiocyanate because it's been known to have an anti-sickling impact. And we'll go a little bit more into some of the research revolving around that later on in our presentation. So the coevolution. So we listened to a little bit of Dr. Fatima, um, um, thanking the creator constantly for her because she's such a jewel and for us to be able to understand this coevolution. Um, before I met her, or not necessarily met her, but heard about her work, we were doing work around it, but being able to have a doctor, somebody working at Howard University, take that information or doing the research around this information and institutionalizing it is a big step forward for the sickle cell community. So while sickle cells are more resistant to the malaria parasite and cassava eating normalizes the shape and the behavior of the red blood cells, this does not increase susceptibility to malaria. And Dr. Fatima Jackson has researched how cassava provides the photochemicals that weaken the malaria parasites. This is an example of what has been called human plant parasite coevolution. So, a lot of, if we did some data or to even took a survey within the sickle cell community here in the States and asked how many of us have eaten cassava before, or if we said, within a week, how many times would you eat cassava, or within a month, um, I think a lot of it, we probably would get information that comes back with less than maybe 10 to 5% of people within the sickle cell community who are using cassava as a staple food. And so this is where that nutritional deficiency comes in. Um, that we have to work towards resolving. The African yam, the moringa, as well as the cassava. These are the things that have to be shifts within our culture as a way that we can eat according to our genetics. So some of the limitations of Western medicine. So our contemporary medical system does not at this point offer considerable portion of time studying methods of nutrition or holistic treatments for the sickle cell community. Um, I've heard time and time again that when doctors are coming through um, their medical school, that they spend they don't spend a considerable amount of time studying sickle cell at all. It's a very small amount of time. That can also kind of be made. That kind of corresponds with how in schools here that we get a month and it's the shortest month for Black History Month. and it's kind of corresponds with how much time they spend on studying sickle cell within the medical schools. So the Black Panther Party, we can learn a great deal from the grassroots organizations that sought to empower their communities and protect their basic rights, such as the Black Panther Party, who in 1971 were the first to acknowledge sickle cell anemia and neglect as a byproduct of imperialism. In 1972, Congress passed the National Sickle Cell Disease Control Act, which for the first time provided authority to establish education, information, screening, testing, counseling, research, and treatment programs. So the Black Panther Party got a negative rap for a very long time because of some of the beliefs that they had and how they chose to move forward or institutionalize those beliefs in the Black community. But this is one of the great things that they did. They put pressure on the federal government and on Congress to add more support to the sickle cell community, and they didn't sit around asking for help. They actually were creating Black community survival programs um, that address some of the needs within the sickle cell community, also offering free testing for people within the Black community who could have sickle cell. So they did a great thing. Not only did they do the sickle cell anemia testing, they also started the programs that would develop into WICs, Women and Children programs, and also they were the first ones to develop what was adopted into the public schooling program, which was the free lunch programs when they started their free breakfast programs for children. So big shout out to the Black Panther Party, because even in our presentations, our understanding about how we can constantly evolve or push forward to culture revolving around sickle cell, we have to first acknowledge that we're standing on the shoulders of giants who came before us to address some of the needs within our community as well. So the Black farmer problem, we touched on it a little bit a little earlier, but food deserts could definitely have an adverse impact on our ability to have staple foods or to cultivate foods specifically for our cultural needs. And so when we look at the inner city, which predominantly, is Black people, where we are urban America, we are in the inner city. A lot of these places that we are pushed into or placed into are food deserts. So we have to be able to understand, well, what does that do intergenerationally when people are not in a capacity to grow their own food on their own land? What happens? Is it a cultural breakdown? What's the phenomenon of that? When we have been for a long time been people who for generations have lived off of the land. When we look at our great grandparents, a lot of them were farmers. So food deserts, as well as the loss of 98% of land associated with Black farmers. That's huge, 98%. Wow, right? So yeah, we're going to have some type of health problems when we have 98% of the land that we used to have stolen from us. Um, Food deserts, as well as the loss of 98% of land associated with Black farmers, has been attributed to the lack of culturally specific foods available to the sickle cell community for the nutrients needed to sustain a holistic health plan. So we have some challenges, but we want to work towards making some of these challenges um, more manageable. So that's what our information platform is developing, these nutrition plans that we can offer to members of the sickle cell community, so we can address some of these nutritional deficiencies. So culturally specific food security. Our disposition here in North America is that the foods that are available in abundance to us are not feeding us on a molecular level. So we have to understand that sickle cell was what they consider to be the first molecular disease discovered. So because it's a molecular disease, we have to also work towards feeding ourselves on a molecular level, eating according to our DNA. This has been engineered over time by capitalistic greed and colonialism with the globalist theme. So the taking of land, the engineering of food to be able to mass produce it. These are all capitalistic models of controlling land and essentially controlling the people on the land. So racism in healthcare. Most of the people who have sickle cell, there's been a trend in them being classified as drug seekers, right? People who need opioids or pain medications because they are in pain. And, it's one thing for them to come to get opioids, and there's another thing for the institution to provide them with opioids or to introduce them to opioids and then stigmatize them when they come back to get more, right? It's, uh, it's a shame. So while there are many sources of stigma experienced by persons with sickle cell disease, racism is an important source as most affected individuals in the US are African American. Despite sickle cell disease being a genetic disease, it is impacted by racism and healthcare equity issues, including hindered access to care and less funding support. So that's something that there's a constant struggle within the sickle cell community to receive the fundings to be able to establish these sickle cell support groups or sickle cell research institutions, right? And so that's one of the things that we have to work towards is being able to get the resources that we need to have the impact that we want to have. So some more of the racism in healthcare. James B. Herrick was the first to discover in 1910 sickle-shaped red blood cells on a blood film of a dental student from Grenada. His description of the student's disease was known for many years as Herrick's syndrome and is now known as sickle cell disease. The condition is prevalent in West Africa and has been known for centuries. So it's interesting how somebody can take blood out of a Black person's body and then classify it through a European's name and have that be how you classify it. It just boggles my mind. So in November 1949, Linus Pauling published Sickle Cell Anemia, a molecular disease, in the journal Science. It was the proof of a human disease being caused by an abnormal protein, and sickle cell anemia became the first understood molecular, first disease understood at the molecular level. So Pauling supported eugenic theory by suggesting that human carriers of defective genes be given a compulsory visible mark, such as a forehead tattoo to discourage potential mates with the same defect in order to reduce the number of babies with diseases such as sickle cell. So these are the people who are, attributed to discovering or being the founders of understanding sickle cell or giving the lens on how we understand sickle cell here in the Western world. And we can see what their attitudes were towards our community, right? A tattoo on your forehead so that people can stay away from you. So we have to move out of this contemporary paradigm so that we can create a new way of addressing our needs, our genetic needs. So cystic fibrosis is a genetic disease that primarily affects individuals that are white and occurs in approximately 30,000 individuals in the US. Is more than 8 times more likely to receive funding than sickle cell disease, despite there being far more people affected by sickle cell disease. So racism frequently interacts and exacerbates the sources of health-related stigma in sickle cell disease, including disease and opioid-based stigmas due to the complex juxtaposition of these concepts. Racism is not only recognized as a source of stigma, but is also seen as a key to stigma. Stigma's possible impact on sickle cell disease outcomes is important to the degree that feelings of shame and guilt reduce psychological well-being, inhibit social engagement, and create difficulties in social roles and interpersonal relationships. So these are some of the things that people within the sickle cell community are dealing with, even outside of their pain crises, even outside of their daily tasks that they have to do. When they're in pain, if they're going to go to school, if they're going to go to work, if they have children, how do they take care of their children? they also have to think about the stigma that comes from having to go inside of um, these healthcare clinics or hospitals that are, because the institutions are founded on racism, that they're stigmatizing them based on the needs that they have, right? And the capacity for them to know what they need is ultimately based on what they've been given in the past from the hospitals. So it just kind of puts them in a tough position. So that's why we're working on nutrition, because we want to minimize some of these negative expressions of our genetics by feeding ourselves on a genetic level.
Mr. Brown, it's 12:05, so this is a good time to take a break. I sent you a message. We got about 5 minutes. So
Okay.
should we break here, or should we finish this slide and then break after this slide, or what do you want to do?
Let me see. Okay, let's do this last slide and then we'll break after this slide. And you said we
Okay.
want a 5 minute?
Yeah, it'll be 5 to 7 minute break.
Okay, perfect. Okay, so individuals living with sickle cell disease who perceive stigma about their condition are at increased risk for negative self-evaluations, loss of self-esteem, anxiety, and social withdrawal, all of which can have a deleterious impact on health, behavior, and health status. So we'll take a 7 minute break and then we'll come back for more of this information.
Okay, awesome. Thanks. Everybody, we're going to take about a 5 minute break, and then we'll come back and we'll do our little warm-up exercises to bring us back to focus, and then we will continue with the presentation. Thank you. Okay, everyone, welcome back. That has been the 5 minutes. So we'll do a little bit of a warm-up session, get us back into the flow of things, and continue with Mr. Brown's presentation. And then we'll have a Q&A section shortly after that, and then that'll be pretty much our day. So first thing I want you guys to do is, if you're sitting down, take a couple of moments to just sit up, stand up off your chair, and then sit right back down onto your chair. Do about 6 reps of that. And then after that, on the final rep, on the 5th rep, I want you to stay standing. And then I want you to have your arms completely extended out at shoulders level. And then I want you guys to bring your arms into your chest and then above your head and then back down and out. So in, so both your hands are touching, and then up above your head. Hopefully everyone can see me. If not, if we stop the screen share, we can see it. But again, we're going here, going up and down. We'll do 10 of those reps. Once we've done 10 of those, guys, then what we're going to do is we're going to do shoulder rolls. So shoulder rolls basically have your chest up, good upright form, good upright posture, and then rotate your shoulder blades backwards for about 10. And then rotate your shoulders forward for about 10. And then we should be sitting back down again. After we've done 10 of everything, we're going to take our heels, put them firmly on the ground. We're going to bring our toes up to our shins and then back down to the ground. These are called little calf / shin raises. And that really gets the blood flowing down into your legs. So do that for about 20 of them. Once you're done with that, take a nice deep breath in. It's 12:15, and we are now finishing the last leg. So go for it, Mr. Brown.
Okay, thank you. Thank you for that. Okay, so community organized solutions.
What can I stop you for 1 second?
Yes.
Is there anybody that has any questions right now for Mr. Brown? Okay.
Okay, so what we want to do is we want to make a positive impact on the life and the well-being of the sickle cell community using sociological tools to understand, intervene, or enhance life for sickle cell warriors. So this was an event that we did. So we organized to assist in the bringing about of solutions to the social problems that developed into alienation and disfranchisement within the sickle cell community. Encourage individuals individual responsibility by teaching the necessary tools to heal and succeed in life with sickle cell. So we have held community events at Sugarloaf Moringa Farm, which was the last slide, to encourage youth and their families from Northern California to learn about farm life, team building, and art as a pathway to entrepreneurship. It was here that we discussed the narrative of sickle cell with community, as well as the differences between growing food within an aquaponics system in comparison to traditional farming techniques. We partner with Bay Area Wilderness Training to offer children and their families tents, sleeping bags, and other outdoor equipment so they could participate in Earth Day and an Earth Day project over the weekend at Sugarloaf Moringa Farm. Our goal was to plant 6,000 moringa trees over 48 hours as a way to support our food equity program for the sickle cell community. Though we did not plant thousands of these trees, we did raise awareness on the need to cultivate culturally specific foods for the sickle cell community. The standard of care that families experience who have children who are overcoming sickle cell is traumatic, to say the least. The contemporary standard of care provided in Western medical institutions has some serious systematic alterations that need to take place. Changes are being made and will continue to be made. What we need to see now is a shift being guided by those directly impacted by sickle cell in collaboration with the physicians, so that the voices are being heard and people aren't being labeled as their disease. We have developed a cause-oriented campaign to support the health and wellness for our sickle cell warriors. This is based on us providing members of the sickle cell community Moringa wellness packs at no cost. This is done by matching sales through our information platform. Currently, we are working towards a solution to the sickle cell problem that is non-invasive and focuses on clean eating as a way to create an inner harmony within the body of our sickle cell warriors that manifests externally. Our conscious of self-healing tools focuses on culturally specific foods as a way to bring about an anti-sickling impact on the blood, using the information provided in this presentation while applying and integrating the science. Sickle cell warriors who are willing to participate in our study for a month will receive our conscious of self-healing tools at no cost, and are encouraged to use this time to collect data on their health improvements before and after. Our efficacy will revolve around a healing paradigm that acknowledges nutritional supplements, digestive health, and right eating to be an alternative to the contemporary invasive techniques that have become synonymous with iron overloads, loss of organs, chemotherapies, and possible loss of life. All in the name of resolving the first discovered molecular disease in the USA. Our method brings the healing of the sickle cell community into the hands of the sickle cell community through food justice, right education, and outreach.
I got a question,
Yes.
Mr. Brown.
Mhm.
The moringa, are you have that on your platform, and
Yes.
that's available? What does it cost, and
It's available online now. It's called the Greenman Pack. It's $20 for 100 capsules. And then for people within the sickle cell community, if you have sickle cell, you can get it at no cost by putting in promo code SC22. And so we make that available to members of the sickle cell community so that they can get used to utilizing these nutritional supplements in such a way that they can become staple foods in their everyday life. And so they're in vegetarian capsules. These capsules can be opened up and put directly into their water, or even put into their oatmeal or their smoothies, or it can just be taken directly as a capsule as well, just drinking it with water.
great, okay, because I want to get some. Okay, so do you
Yeah,
have you're gonna have that information up for people to see what you just told me about?
Yeah, so I got a little bit more that I want to go through in the
Sure.
presentation.
Sure.
Yes,
I
I'm
just,
going to touch on a few more things as well.
okay, that's fine. I just wanted to make certain that everybody gets this information because I want it. And if you had
Okay.
a slide with it, then that would be great.
Okay, I'm going to go over that now since you brought it to
Okay.
my attention.
Okay.
Okay, so I'm going to share this page here. So, share. Okay, so here we're on the information platform. And if we go to Moringa here, so if you go to creationofsociety.com, it's you know, at the top is one of the tabs are going to be Moringa. And so once we click on Moringa, it will take us here, and then the agreement pack here. You click on this. Okay, and so it says $40 on here, but you would have to select how many months you want. So if you go for 1 month, it's going to go to $20. And if you wanted to go for, you know, 5 months, then it's be $100. And then that subscription will just basically be how many packages that you wanted. So you can go anywhere from 5, which would be enough for 5 months, or you can go for 2 months, 3 months, or 4 months. And so if you wanted to add this to your cart, and then when you went through the process of adding it to your cart, once you put in SC22, what that would do is you would put your information in, your mailing, and your, you know, your shipping address, and then it would be no cost, you know, for members of the sickle cell community. And so of course we have more, you know, different products on here. We have the Moringa, which we also have the Moringa sea moss, which is a blend between the two superfoods. We have the Moringa oil, which is very good, you know, just as far as this is like a topical technology that prevents airborne impurities from entering into the body through the skin. It's also good for the elasticity of the skin as well. And it's been used for centuries as an anti-aging oil. They've even found these in the tombs of the pharaohs to keep their skin pretty much intact for thousands of years. We have the yam vitamin, this is one of the tubers that was discussed with Dr. Fatima. While this isn't necessarily cassava, this is the yam vitamin, which has the highest concentration of the isothiocyanate that we're going to touch on now a little bit more in detail. I want to bring us to an article that was put together in 2008. And so the diascovite, this has been on the market for over 30 years, but a lot of us have never heard of it before. And a lot of that has been very strategic, you know, as far as keeping, you know, certain information outside of the scope. So,
Christen, I had a quick question on, you just brought something in that I wanted to ask about. Why did you guys decide to go with Moringa versus the cassava, like Dr.?
Well, you know, the cassava is the regions that it's grown in, they have a different climate than we have here in California. And so the Moringa, it can be grown here in California, which allows us to be able to kind of control that process a little bit more. And it still is a good source of the isothiocyanate. So while we may be importing things like the African yam or the cassava, growing the Moringa will actually allow for, you know, someone like ourselves who has a child who has sickle cell to be able to go right outside and have that relationship with the cultivation of the plant. So they'll be able to take the leaves off, eat the leaves, you know, dry the leaves out, and they're developing that coevolution, that relationship with the tree that will be working towards sustaining their health. We didn't have the same opportunity to do that with the African yam here in California, or the cassava. So what we did is we chose this particular superfood to be not necessarily a substitute, but an alternative for that. Make sense?
Yeah, and so you kind of get the same nutrients, or is it different nutrients? Like, are the side effects different at all?
No, there's no side effects. So, you know, the photochemicals that were discussed with Dr. Fatima, we still get those. So let me go into some of the benefits of the moringa as well. And so really the reason why we chose it, because we're able to still get that isothiocyanate, of course. We're able to get the chlorophyll, which is very essential to blood production as well. We're able to get the iron. The iron richness is still there. So we have a lot of good benefits, huge benefits that come from the moringa, that makes it extremely beneficial. So I want to, before we touch on that
Okay,
document-
yeah, don't move from that only because, I'm sorry, I just want to, right here in this, this slide that you have, it says objectives. I can't pronounce that, but hydroxyurea is the next thing. So how does that come in? I mean, what is, you know, because all of they give everyone living with sickle cell, at least the medical.
Okay. So what I'll do is I'll touch on this one first, and then Taurus, I want to answer your question after I go through this one, okay? And so we'll
that's
touch on
fine.
moringa and some of the benefits of it, but I do want to discuss this comparative study from Pakistan.
Okay.
So it says a comparative study on the anti-sickling properties of thiocyanate, tellurite, and hydroxyurea. So a lot of us have heard of hydroxyurea, right? As just the basics, you know, for sickle cell.
Right.
Well, hydroxyurea didn't necessarily start off as something specifically for sickle cell. It will start off as something specifically for chemotherapy patients. But thiocyanate is something so richly abundant in nature that there's, you know, you can't really control it. You know, there's a family called the Brassica family, and the entire Brassica family is a rich source of thiocyanate. And there's over 500 different plants that carry this thiocyanate, which means that this is something that's not going to be controlled. You know, it's just being able to have the information available to be able to consume it as a staple food. Okay, so I'm going to read through this. It should take a few minutes. So the objective here, thiocyanate, hydroxyurea, and tellurite are among chemical agents being used as anti-sickling drugs and currently receiving attention for research. The anti-sickling properties of these drugs was investigated and compared in this study. So the methodology, the human sickle blood was incubated in the drugs in vitro at concentrations related to the dose used by patients in vivo. Hemoglobin function and specific aspects of the sickling process were then measured by employing standard methods used in screening potential anti-sickling agents. The results, all the drugs significantly inhibited sickling of deoxygenated sickle blood and formation of irreversibly sickle cells in a dose and time-dependent manner. Thiocyanate, hydroxyurea, and tellurite inhibited optimally 20 mm, 40 mm, and 50 respectively. Okay. Thiocyanate and hydroxyurea prolong sickle red blood cell lifespan, as indicated in the significant decrease in homolysis and osmotic fragility, while tellurite increased these blood parameters. The three drugs also cause significant prolongation of delay time of hemoglobin S, polymerization, while thiocyanate and hydroxyurea significantly increase both solubility ratio and oxygen affinity. Solubility is basically saying that the blood cell isn't crystallizing because it's dehydrated, it's still soluble. So conclusion, the results obtained in this study suggest that the three drugs have remarkable anti-sickling potential in vitro, with thiocyanate being the most efficient, followed by tellurite. So hydroxyurea didn't make the top two. Sickle cell disease is a genetic blood, so we kind of know about that part. So we'll go a little bit deeper into this. Some promising anti-sickling earlier investigated cause homolysis of sickle red blood cell at their effective dose levels and therefore unsuitable for clinical use. Hydroxyurea, tellurite, and thiocyanate are among the long list of chemical agents currently being used as anti-sickling drugs and undergoing intensive studies. The present study intended to compare the anti-sickling actions of these drugs and their effects on selected red blood cell parameters so as to further understand the mechanism of action. So the materials and methods, the human sickle cell blood obtained from patients who attended regular clinic at the hematology department, the University of Iran teaching.
I don't think you need to go over materials and methods. I think you just need to go over the results and the conclusions, just so that we can understand why those two actually have more anti-sickling properties than the hydroxyurea. The problem, most of the problem sometimes is just finding adequate amounts of those resources. So I thought maybe this study was going to talk about more of that. Because it's known that those actually do have more anti-sickling effects. But the problem is, at least from what I know, from my current knowledge within the scientific field, is just finding enough of a source for that. So
okay. Well, for the second time, let's jump right into the results. The optimum sickling inhibitory concentration for thiocyanate, hydroxyurea, and tellurite was found to be 20 mm, 40 mm, and 50 UM respectively. Okay. These are the minimum concentrations required to achieve maximum inhibition of sickling within 1 hour. So they have the time, show the time-dependent inhibition of sickling by 3 anti-sickling agents. Blood incubated with thiocyanate had the least number of sickle cells compared to tellurite and hydroxyurea after 2 hours. So I can read that again. So the blood incubated with thiocyanate had the least number of sickle cells, which was 38%, compared to tellurite, which was 50%, and hydroxyurea was 63%, right? After 2 hours. Untreated sample had 93% sickle blood at the end of the incubation period. With respect to the inhibitory effect of the anti-sickling agents on ISC formation. Figure 2, the percentage was reduced from 28% in the untreated blood to 14.1 in thiocyanate blood. In tellurite treated blood, 19.5 in hydroxyurea treated blood after 2 hours and 30 minutes of incubation. Okay, so these are just different charts that if you want to come to the page and see at any time, they're going to always be available. Thiocyanate and hydroxyurea significantly increase percent of oxyhemoglobin by 101% and 39.0% respectively. So thiocyanate percent increased it by 101% and hydroxyurea increased it by 39%. Compared to the control of compared with the control, while oxyhemoglobin content was not affected by treatment with tellurite. In addition, tellurite and hydroxyurea significantly stimulated the methemoglobin production by 61.5% and 45% respectively compared to the control. Hemoglobin solubility was significantly enhanced in the presence of thiocyanate by 130% and hydroxyurea by 54% compared with the control, while tellurite did not alter the solubility at all. all the drugs cause significant prolongation of delay time in hemoglobin sickling, polymerization. Tellurite was 156%, thiocyanate 114%, and hydroxyurea 71%. Compare with the control as seen in table 1. So the discussion in vitro, the abnormal shape of the sickle ethrocyanate in the deoxygenated state was markedly improved, as shown by significant reduction in percentage sickle cell upon incubation with the drugs. The sickling inhibition was found to be dose and time dependent. Thiocyanate appears to be the most efficient among the 3 anti-sickling drugs in vitro, as the blood incubated with thiocyanate had the least number of sickle cells at the end of the incubation period. This might be because thiocyanate has a different covalent modification effect on the hemoglobin. Thiocyanate reacts with the amino terminal valine residue of hemoglobin and carbulated. While hemoglobin S is carbulated, it is maintained in the R form, which is not susceptible to sickling. So it's more information. We don't necessarily have to read all of it right now, but this is just showing you that there is research being done on a global scale revolving around thiocyanate in comparison to some of the other drugs that are on the market. I wouldn't necessarily consider thiocyanate to be a drug. I will understand it more of a nutritional supplement because it's already found available in foods already.
Thank you.
No problem.
And that information will be there so everybody can refer to it at a later point in time. I appreciate it. Thank you.
So this is, if you go to the blog and you go to comparisons on sickle cell from February the 17th, all that information is available there. I want to touch on your question, Taurus, as far as the moringa, just so I can show you some of the nutrient denseness of it as far as why we chose it. Chose to use this in comparison to cassava, which there isn't really any traces of it being grown here in California. So moringa, when you think about antioxidants, so let's go up a little higher.
So you could correct me if I'm wrong, but basically you guys decided to go with moringa not only because it's a quick, because from what Dr. I keep forgetting her name, what she mentioned was the fact that the cassava is the most highest in thiocyanine richness, right? But then you guys decided to choose moringa because it was easier to grow in California. Why is it so important that that relationship be established versus just importing cassava?
Well, you can always get cassava, like it's already imported, right? So if you go to any export African store market, you can pick up cassava and you can pick up the African yam. So it's not that we're not using it; we still have access to it, to the availability of it. Nor
well, no, I didn't say that you're not using it. I'm just saying, like, what? Because you just said the importance of it, of why you chose moringa is because it could be grown in California. So I'm just curious as to why that's such a huge factor when cassava has actually been proven to be more effective.
Well, it's not that it was more effective. It was used in that region. So when she was developing her research, you know, while they may not have been growing moringa there, she was able to identify the certain plants that they were using that was having an anti-sickling impact on the blood. And in that region, it was cassava. Now, if you go to Nigeria, it may be the African yam, and they could still be having an anti-sickling impact on the blood. Now, if you go to Southeast Asia, where moringa is grown abundantly, you know, it's been known that Southeast Asia might have the highest concentration of sickle cell on the planet, right? And they may be actually using the moringa because it actually grows in India as a part of their Ayurvedic medical system. And so that might, in that region, it might actually be decreasing some of the negative expressions that that can come from sickle cell anemia. Our choice to use moringa was
the highest concentration of sickle cell incidences and diseases in India, Southeast Asia. I thought
Yeah.
it was in Africa.
No, that's a lot of some of the misconceptions that revolve around it. There's a larger percentage of sickle cell cases, not necessarily in Africa, but um, in Southeast Asia.
Huh.
Yeah. So we chose to use the moringa in collaboration, right? So it was a part of us developing a nutrition plan. It wasn't necessarily um, saying that this is going to be the only thing that we're going to use, but because we chose to be vegetarians, we had to include more than just one particular plant. So while we are using the yams and while we are using the cassava, we're also using the moringa, while we're also using other, um, other herbs and things like that. But if you look at, when we did our study on, are you familiar with moringa at all?
No, I'm more familiar with cassava, which is why I'm asking the question.
Okay. So when we think about the antibacterial, the antifungal benefits of moringa, so these are the things that can help keep people healthy, right? Antifungal, antibacterial. So in 1964, as a PhD thesis, Benny, student of the well-known chemist, identified a number of glucosylated derivatives of benzoyl isothiocyanate, and so that's what we were discussing a little earlier. Compounds containing the 6-carbon simple sugar renus. The identity of these compounds was not available in the referred scientific literature until rediscovered 15 years later by Kajar and co-workers. Seminole reported on the antibiotic activity of the primary rensylated compound, then followed by his colleagues. The re-isolated and confirmed the identity of these benzoyl glucosinates and cogenate thiocyanate, and verified the activity of the later compound against a wide range of bacteria and fungus. So those are things that it's eating every single day. What it's doing is just strengthening the immune system, right? The omnipresence pathogen of human beings in medically underserved areas of the world amongst the poorest of people populations, a major cause of gastritis and gastric ulcers. So let's just go, so it's cancer prevention. So I want to go into the key nutrients in moringa and disease prevention. So looking at what we have here, 3 times the potassium in bananas, 3 times the iron of spinach, 2 times the protein of yogurt, 7 times the vitamin C of oranges, 4 times the vitamin A of carrot, and 4 times the calcium of milk. So by providing abundant vitamin A, moringa, so these are, when we think about it in comparison to some of the traditional staple foods are, we have huge gains in the moringa. Being able to cultivate it here in California, we still get all these benefits. So when we think about iron, by providing abundant iron, moringa helps prevent anemia, fatigue, irritability, weakness, shortness of breath, dizziness, pale skin, headache, decreased appetite, especially in children, and brittle nails. So iron deficiency is one of the most common known forms of nutritional deficiencies, and that's anemia. In the human body, iron is present in all cells and several vital functions as a carrier of oxygen to the tissues from the lungs in the form of hemoglobin, as a transport medium for electrons within the cells is the form cytochromes, and as an integral part of enzyme reactions in various tissues. Too little iron can interfere with these vital functions and lead to morbidity and death. So we got an iron-rich food here in moringa. And so with the consumption of it every single day, we get the iron richness, and we also get that isothiocyanate as well. But if you do have time, go look over that interview or the presentation by Fatima Jackson because she actually never mentioned isothiocyanate in her presentation. But she did mention that the cassava can have an anti-sickling impact on the blood. So while we're focusing more on the
Well, she was talking
isothiocyanate,
about the effects of cyanide because you can get well, she thought that you could get cyanide poisoning from eating whole cassava because normally it gets broken down because of the, that's why she was talking about cherry pits and all that stuff. So
yeah,
um
And I had a question.
yeah.
yes, please.
Okay, so like, if this is really rich in iron and everything, to help the blood cell with the production of anemia, to stop anemia, wouldn't it be bad for a person to take this drug or supplement? I'm sorry, not drug, supplement, if they're iron overloaded?
Well, if you're iron overloading, it's not because you're eating a lot of iron-rich foods, right?
No, you're iron overloaded from blood transfusions, but when you are iron overloaded, they tell you not to eat a lot of iron-rich foods.
Yeah, but so without me being a doctor, I can say if you are taking transfusions and it's sending you into iron overload, and then you're told not to eat iron-rich foods, if we were to spin that one time, would the eating of iron-rich foods prevent the need from getting blood transfusions?
No, because you're getting blood transfusions simply because your hemoglobin is low, not because of iron.
But so the iron and the hemoglobin, that relationship is
Hand
those
in
are,
hand. But
yeah.
it's unfortunate that you do get iron overload from receiving blood transfusions, because you can be on blood transfusions not just from your hemoglobin dropping, but in my experience, I'm on blood transfusions to lower my S level and to keep my bypasses in my head open.
You said your bypasses in your what?
My head.
Okay.
I've had
So
surgery, which made me have to get bypasses because of my blood vessels closing out because of the moya. And so that is one way that also prevents, because I can't have a lot of NSAIDs, blood thinners.
mhm.
It helps keep the blood transfusions, because I get exchange blood transfusions. I don't want to say blood transfusions because I don't get regular blood transfusions. I get exchange blood transfusions, which also helps me keep my bypasses open because of the pressure within the blood transfusions.
Okay. Okay. So your question was, if this is iron rich, why would I advocate for someone within the sickle cell community to eat it?
No, I didn't say that. I said a person with iron overload. Why it
So
wouldn't be
what do
healthy
they
for them.
what happens when you are in iron overload? What does the doctor suggest?
When you are in iron overload, they suggest you take iron chelators, which removes the iron from the body.
Okay. And then so when you're no longer in iron overload, is there a suggestion to eat iron rich foods, or is it just to
Yes,
come?
it is a suggestion, because if you don't have iron overload, they definitely suggest you to eat iron-rich foods because you have
Okay.
anemia.
Okay.
But you have a different type of anemia when you're iron overloaded.
Okay. So for my recommendation as someone who uses moringa and has a child who has sickle cell who also uses moringa, I would say maybe wait until you're not in iron overload. And then that can give you an opportunity to use the moringa, and then you can see what the beneficial effects of it are. But I will also advocate for you to be using the isothiocyanate that we have available as well. And if you're using that daily, then what you can do is you'll be able to measure what your hemoglobin level is. And then also, if you're still having the same amount of sickling that you were having prior to using it, because it sounds like you've been going through a lot as far as managing your health. Within the hospitals, they've done you sound like you've been through a fair amount of procedures.
Well,
Um,
I've had a fair amount of procedures, but I also have gotten my body healthy to the point where I'm not in pain
That's
with
wonderful.
the foods that I'm eating, or not having as many complications. But everybody who has sickle cell know that sometimes it can't be prevented.
That's right.
Sometimes things happen, you know what I mean?
Mhm.
And yeah, things happen, unfortunately. But like
Mhm.
I said, I've got myself to the point where I don't have a lot of pain, where I'm taking liquid chlorophyll, I'm taking more supplements than
That's
drugs
wonderful.
and things like that. Like, you know what I
Yes, you
mean?
are
But
taking liquid chlorophyll.
I take liquid chlorophyll every day for my energy levels.
That's wonderful. That's wonderful. That's a good and you see the difference.
Yeah, it's a
And
big
so
difference.
those are some of the photochemicals that we were discussing a little earlier. Is that all these things that the plants are producing? The chlorophyll and the hemoglobin, they're so much identical. It's like the chlorophyll was made for us to use every day. And so those are
Right.
some of the photochemicals that we're also getting from the moringa as well. But you can get it from
Right.
kale, you can get it from cabbage, you can get it from pretty much any
Right.
green leaf.
And I
You know,
also want to say that even although I do take liquid chlorophyll, when my hemoglobin is really low, it's the liquid chlorophyll doesn't really help. I mean,
well,
it helps to its extinct. But if I'm low, really low, it's like well, for me, I just stop eating meat and I do a whole lot of things, but sometimes there's like nothing I can do
well, you need to start doing the
but get a blood
thiocyanate.
transfusion.
You need to start doing the thiocyanate. And that's a lot of it is the reason why we're having this conversation now, and while we've been going over some of the things that been going over, is that people within the sickle cell community, especially just our community in general, this is a molecule that we're not getting enough of to be able to manage and sustain our health. And this is something that we should be using every single day, to prevent these crises and also to prevent the cells from sickling altogether. So when we're using this thiocyanate in such a way that it becomes a staple food, it's getting into our bloodstream. And when it's in our bloodstream, then it's able to have those type of impacts that prevent the cells from sickling altogether. And so that was the goal of this conversation, to bring light to the benefit of the thiocyanate, in such a way that we can acknowledge the need for us to use it every day, right?
Correct.
So that might be the missing link, because if it's something that you're not using or you haven't used in the past, it may be something that your body may be actually yearning for. Make
Well,
sense?
there's supposed to be a lot
Ron
of
has a question.
thiocyanate. The question that I was asking was more of the iron overload in the moringa.
Mm-hmm.
Yeah.
Yeah. So the moringa, it's a source of thiocyanate. It's a source of not just iron, but
But
it's
it
a
wouldn't
source of,
be healthy for me to eat it. You said
well,
just to
it may
wait.
not be healthy for you to eat it when you're experiencing iron overload, because
Right.
it's already been suggested for you to stay away from iron rich foods when you're in iron overload. But the time that you're not in iron overload, my suggestion from somebody who's been using it and who has a child who has sickle cell is that might be something that you want to also implement into your nutrition plan.
Right.
Okay, I'm gonna jump in real quick. I'm sorry, Calvin A. I do appreciate your perspectives on all of this for Calvin A and Kristen. I do want to say this, because the point I was trying to make in chat is that definitely what Calvin A is saying about iron overload, but also I have hemoglobin SC, and I cannot consume too much iron in any capacity because it does cause more pain and sickling episodes. So
Yeah.
just a caution to everyone to be very mindful, because it is going to manifest differently for everyone.
Exactly.
Like for me, for example, I have to go in for medical phlebotomies every 4 to 6 weeks if I'm not in an anemic state. So it's actually like the opposite for the average sickle cell patient, if you will. So just be mindful, because this is not medical advice. And I know SCAASF has their disclaimer on the presenters and whatnot, but just be mindful that it is going to vary from patient to patient. And so it may not manifest in the same way for everyone, and be careful with that.
Okay. Thank you for bringing that to my attention, because I know there are different variations. Everybody's experiencing different things, right? We have different journeys that we all walk on. And you know what may be beneficial for one person, or the experiences of one person, may be something totally different. You know, my son, he's 10 years old now, and he hasn't been hospitalized in 6 years, and that might be very different than a lot of the things that people within the sickle cell community do experience. And I have to acknowledge that people do experience different things. And you know, for someone to start doing or implementing different changes in their life once they become an adult, it's a little bit more challenging to do, opposed to you have parents that are implementing these strategies when you're a child. So I do acknowledge that there are some differences, but the goal of this conversation, for the most part, is to introduce this concept of nutritional thiocyanate as a way that we can control the sickling of the cells altogether, as a nutritional supplement that can be used. The moringa is something that, whether you want to use it or you don't want to use it, you know, it's totally up to the individual. Like I said, it's a good source of thiocyanate. You can also get the seeds, and the seeds, they're not necessarily full of iron, but they still have that source of isothiocyanates. So the highest concentration of the iron within the moringa come from the dry leaves. And so that's what is in the capsules, and that's what will be growing on the tree. But if you were to take the seeds are also a higher concentration of the isothiocyanate, and they don't have any iron in them at all. So the daily consumption of the seeds may be a better approach to being able to utilize the moringa within the sickle cell community if you are experiencing an iron overload. They have the same good source of isothiocyanate, but no impact on the iron level at all. So that might be a better approach. Does that make sense?
Yeah, it makes sense to me.
Okay. And then also we have the supplement, which is a concentration of potassium thiocyanate. And so this doesn't have any iron in it at all, and it's in a liquid form. And so the goal there is to be able to have a daily intake of this thiocyanate in a liquid form that can be used just in the same capacity that someone would be using liquid chlorophyll. So we do have some flexibility around our nutritional supplements that can be beneficial for the sickle cell community, no matter what path you're walking as far as having the sickle cell SS or the SC. These products have been engineered in such a way that they're beneficial for our blood and for our immune system. Any
Okay,
other
so
questions?
no more questions. So Christen, you want to move on with your info?
Yeah, so we have this calcium situation here, where we have people within the sickle cell community that experience the bone deterioration. And that starts to, it happens in the hips, it happens in different aspects of the
Yep.
bones.
All of
And
them.
then so when we think about moringa, as it also can be used for iron, for helping stimulate the iron. It can also be a source of the isothiocyanate. It also has 17 times the calcium of milk. And so when we think about osteoporosis or bone weakness and damage, this is a good source. This is a good source of strengthening those bones. So, you don't have 12-year-old children going and get hip replacements, right? All of it is sourced through food. And so this is another reason why, I become an advocate of the moringa, because it's very versatile. It has a lot of different benefits for it. So calcium deficiency is a major concern in the United States. An estimated 44 to 87% of Americans don't get enough. An easy way to think of calcium and bone health is to imagine the bones as a savings account at a bank. You see, until the ages of 30 to 35, the body stores calcium in the bones. However, after this, calcium is no longer stored in bones. As a result, when the body tries to remodel bones, its only source of calcium is ingested calcium. If you're not eating enough calcium, the body has nothing to use. Thus, the body cannot remodel the bones, and bone density subsequently decreases. The calcium you consume early in life is deposited into your bone saving account. If little calcium is saved, then there will be little to spend when retirement comes. So calcium is a very essential function of being able to have our bone density in check, and not having to rely on these different type of, hip replacements or bone situations, right? So moringa is also a good source of not only calcium, but also boron. And boron is also something that that works on strengthening the bones and increasing bone density. Um, protein is very essential. Um, one episode that we had with our son is I think he might have been 2 years old. He got a blood transfusion because he was experiencing acute chest syndrome. And once he got his transfusion, maybe about 3 weeks later, his hair started falling out. The total texture of his hair went from, curly and kinky to very straight. And people was asking me, did I give my son a perm? I said, my son is 2 years old. Why would I give him a perm? But it was some of the consequences that came from him getting a transfusion, and he started losing some of his hair as well. So I just say that to say, if you're getting these monthly transfusions, you might experience some hair thinning or something, of that nature. And as I don't advocate for the transfusions, I will say that, a healthy diet can, produce the cells from sickling, and then you can also get some of those natural proteins and amino acids that the body needs. Um, so this is 9 times the protein that we have in yogurt, and it can reduce the pigmentation on the hair and scalp and body, reduce skin rashes, dryness, flakiness, general weakness, and lethargy. That's something that, anemia is also, causes that general weakness and lethargy as well. And so I have all this information online as well, and this is free. So you can go to the online store and you can pick this information up at no cost to yourself, and it's a digital download. So moringa trees are known to overcome protein deficiency in developing countries, as the leaves and other parts of the tree is comparable to the soybean, high amount of crude protein and amino acids. So potassium, we've always eaten bananas as a source of potassium, but this moringa has 15 times the potassium of bananas. So by providing potassium, moringa helps prevent fatigue, problems with muscular weakness, skin and related, such as blistering, skin eruptions, dryness of the skin, temporary memory loss, or problems such as weak memory, heart-related problems such as deterioration. So that's huge.
Real quick on the question. So is the powder more concentrated? Is that why it's 15 times more potassium?
Yes. So when you have the powder, it's the leaf is dried out, so it's more nutrient dense. If you were to pick a raw leaf off of the tree, a lot of that leaf is going to carry a high amount of water in the leaf. So it's not going to be as nutrient dense. So if you had, let's say, 1 pound of fresh moringa leaf, it's not going to be as nutrient dense than if you had 1 pound of dried moringa leaf, because all the water has left out of the dry moringa leaf and is more concentrated and more nutrient dense at that point. Make sense?
Well, yeah, and there's also more leaves, right? Because it's dehydrated, so you can fit more into it.
There you go. So they talked about it being used, moringa being used for diabetes. So moringa oleifera is a nutrient plant that can help to maintain normal blood sugar levels. So it holds a lot of promise for our diabetic community as well. This is the primary because of the amazing nutritional benefits. Moringa oleifera has also been shown to possess many key anti-inflammatory benefits. Also causes diabetes, circularity problems, which can be managed through anti-inflammatory supplements. There are no negative side effects associated with Moringa aloe use, meaning that it is a safe, natural way for people to manage their blood sugar and care for their diabetes symptoms. It's just one more option for the many people who have to cope with this serious condition. Um, so antioxidants is huge. Moringa has said to have approximately 46 antioxidants and is one of the most powerful sources of natural antioxidants. Antioxidants supply the free atoms needed by the human body to mitigate and affect of free radicals. Moringa leaves are rich in flavonoids, a class of antioxidants. The beta carotene present in moringa leaves also act as antioxidants. The antioxidants will have the maximum impact on the damage causing free radicals only when it is ingested in combination with nutrients and a group of antioxidants. A combination of antioxidants is more effective than a single antioxidant on an equal weight basis due to the antioxidant cascade mechanism. This is why moringa tea acts as an effective source of antioxidants than any other herbal tea or even a green tea. So it's also something that you can consume the tea, as a meal or as an additive, but it's also something that you can use as a tea as well. So moringa and zeaxanthin. Moringa is jammed with cytokinin called zeaxanthin. Um, Cytokinins are plant hormones that help cells divide and protect against oxidation. Zeaxanthin is the most powerful of all cytokines, according to the 2004 Danish study. Zeaxanthin helps promote small cell size as a key component to more youthful skin. It also influences the structure and functional integrity of the cell and prevents accumulation of macromolecule damage in the cell. And study found that zeaxanthin increases the activity of some antioxidant enzymes, counteracting the free radical induced oxidative damage incurred during cell aging. And so this is a lot of information on here. We don't necessarily have to go over all of it now, but like I said, this is free information that you can find on the information platform, and you can download it whenever you like. It's going to be available. From here, I actually want to go back to the platform, and I have some more information that I want to show you there as well. There's always going to be times in life where you got to rise up out of. Okay, so this is, so is anybody familiar with Linus Pauling? Okay, so I want to show this here. It's 2 minutes. And so Linus Pauling was attributed with the discovery of sickle cell as a molecular disease. He was given Nobel Prizes. He also has a college named after him at Oregon University, where they focus a lot on isothiocyanates there, as well as dietary supplements. But this is also the person who had this eugenics idea as far as the tattoos on the head of people within the sickle cell community. But I just wanted to give you, some insight on some of the thinking behind how he arrived at some of these perspectives.
Along with this protein study, he had also been spending years looking into the properties of hemoglobin in blood cells. Normal blood cells like these are disc-shaped, which enable them to pass through blood vessels easily. But blood cells can be diseased. Sickle cell anemia results from the sickling of cells so that they become rigid and crescent-shaped. The sickled cells clump together, making it more difficult for them to pass through the blood vessels. The disease is inherited and often causes pain and weakness in its victims.
I had the idea in 1945 that the disease sickle cell anemia might be the disease of the hemoglobin molecule. No one had ever, so far as I'm aware, no one had ever suggested the idea of a molecular disease before. As soon as I had this idea, I thought it must be right. From what I know of the properties of these patients, I believe that this is a disease of the molecule, and that if we look at the blood of these patients, we shall find that the hemoglobin molecules are different from those of other people.
Oh, yeah. Pauling had made a profound, educated guess, but it took years before techniques were developed that could investigate his ideas. Eventually, it was confirmed that the victims inherit hemoglobin, which does indeed have a defective molecular structure.
What is happening with that?
Dr. Harvey Utano, who began working with Pauling in 1946, is today, along with other scientists, experimenting with different chemicals, trying to stop blood cells from sickling. Pauling has taught a generation of scientists to think in molecular terms, and he helped to establish the new science of molecular biology.
Okay, so when I think about a lot of the work that is being done around sickle cell, I also had to take into consideration with some of the origins of how we understand sickle cell and who is responsible for developing our thinking around sickle cell. And so when I think about Linus Pauling's contributions to us identifying and understanding our own genetic material, I have to also understand that there is some gray area with the work that he's done, who the work was impacting, and then what are some of the other cultural phenomenons that our community has gone through that could cause some type of deterioration on a molecular level. What were some of the challenges that our community has faced that may be overlooked and within the scientific community that is still relevant to how our families function as sovereign human beings. From here, I want to, this is another 2-minute video. It was an interview that I did with a sickle cell warrior, and she's talking about some of the challenges that she faced and some of the practices as well, as somebody who has sickle cell.
My name is Tiara. I'm 27 years old. I was diagnosed with sickle cell anemia when I was 10 months old. My parents did not know anything about sickle cell. They didn't know, they never even heard of it, didn't take blood testing for it. I got really sick, ended up with pneumonia, went to the hospital, they did blood testing and found out I had sickle cell. So from that point on, it's been nonstop hospital visits. I probably had over 500 hospital visits in my 27 years of life. I had a stroke when I was 7. I had my left lung collapse when I was 10. I used from the age of 4 up until I was 18. I had MRIs every 6 months to make sure that I wasn't going to have a stroke or another stroke, even though I did have one. When I was 17, I had my gallbladder removed because I had gallstones. When I was 14, I was diagnosed with something called avascular necrosis, where basically my bones in my hip and my knee started to deteriorate because there was no oxygen, no blood flow to that area. And so over the time, I just had no bone. It was just bone on bone rubbing. And I ended up having a hip replacement at the age of 24. I will be having a knee replacement probably before I'm 40 because of the avascular necrosis. I am monitoring my sickle cell carefully now, sleeping, no stress, a lot of water. The pain is every day. I probably live at a 5 every day, but I get up and I get through it. And I do take hydroxyurea, which is the chemo medicine. I've been taking it since I was 12. I haven't had much success on it, but my doctors feel like that's the only way to go right now. And basically, I haven't had a hospitalization since May, when the last 4 years I've been hospitalized every single month. I do have a port here in my chest because every time I go into the hospital, I have no vein access, neck, feet, head, anywhere, there's no vein access. So.
That's deep. That's deep. So that is just, that is some of the that's just what people within the sickle cell, the warriors, are faced with, right? And a lot of our family within the sickle cell community, because the first time they hear about sickle cell is in the hospital, so there's no other way to approach it than the hospital, right? Because that's where the information is developed at. So our goal is just to broaden that scope some more. I'm not saying that you don't utilize those resources that you have within the hospital, but we also have to be in our nutrition bag. We have to be able to figure out, well, if there is a situation where the bones are deteriorating, are there certain plants that work in such a way that if we consume them every day, that after 6 months we can see some type of reversal happening? If the cells are sickling, is there a certain food or a certain plant that we could be utilizing that after 3 months that we'll be able to see improvement as we use it as a daily staple? And I know that that's out there. And so the information that I want to provide you with, anytime that you want to have more information on this, that's why this is set up to be that for us. It's set up to be an information platform so that we can have this information available to us, and so we don't have to constantly be discussing the negatives, but we can also be building upon what some of the solutions are going to be for our sickle cell community, right? And like Dr. Fatima Jackson discussed, we have to build upon this concept of plant-human parasite coevolution. So these plants are going to be evolving our circumstances the more that we evolve our understanding of the plants altogether. So we're advocating for the use of the thiocyanate, which we have that product available online called diascovite. It's an extract from the African yam. We also have moringa, which, if you're not experiencing iron overload, it would be a great daily supplement to utilize. One for the richness in iron, but also for this richness in calcium, also for its richness in potassium and protein. And also, if you are experiencing iron overload and you still want to use the moringa, you can eat the seeds instead of eating the leaves. The diascovite is available online, and we also have the moringa sea moss, which is available online. The moringa sea moss is $35. It has 100 capsules that come in vegetarian. They're encapsulated in vegetarian capsules. Sometimes you see these capsules and they're encapsulated in gelatin, which is animal cartilage. Because we're vegetarians, we try to make sure that everything that we're using is plant-based. We also have the moringa oil is available as well. So we want to be a space where we can broaden, through our experiences and through our journeys, that we can broaden our capacity for being able to understand how to approach our genetic health using plants as a step in the right direction. I'd like to open this up for any more questions or comments or anything that I may have missed that I could have touched on that you may want me to touch on now.
I just have one question. In terms of that term, plant-human parasitic evolution, did Dr. Jackson coin that term, or who came up with that term?
Dr. Fatima was the first person I ever heard of develop this concept of human-plant parasite coevolution.
Interesting, because I would think it would be more of a symbiosis, but I guess that's a question for her. Because parasitic basically means that one organism is taking over another organism, or basically depleting the other organism. of its resources. But it seems like this is more of a symbiosis where both are kind of developing at the same time and giving each other benefits. But I mean, I could be wrong.
No,
so.
no, you're right. I think you're absolutely right. There's an echo.
Yeah, I think somebody's unmuted. Let me mute everybody. Hold on. All right, I think the echo's gone now.
Okay, I think you're right. You know, within the relationship between plants and people, right? So if we look at the cassava or the African yam or the moringa, this is a symbiotic relationship. This is people cultivating a plant and the plant working to benefit the people's health, right? But the parasite aspect of it comes in with malaria, with this genetic phenomenon that's taken place, because the parasite was actually how the malaria was being transferred to the people through the mosquito. So that was the concept of people plant parasite coevolution, because the mosquito may still actually, you know, bite the person or take the blood from a person, but the opportunity for that malaria parasite to cause dysfunction inside the body, when it's met with the sickle cell, it doesn't have that opportunity anymore. But when that cell sickles, the coevolution happens when the food is eaten, that cell can go back to a whole shape, if that makes sense to you.
It only makes sense if the evolution were apart from. So if the evolution were a direct effect, but actually, the way that evolution works, at least from my perspective, or from what I've learned and know, is that, you know, a bunch of people were living in an area and certain traits were taken and other traits weren't. And one of the traits that were prevalent in places like Africa and that region, because of the existence of malaria, was the sickling, which is why sickling even exists. The only thing that the plant would actually help with evolutionarily is what you just mentioned, is it has certain properties that would help people with being able to combat the sickling, but the sickling is not a direct effect of being bitten. And I think it's just the fact that malaria was already existent, and those people that were actually resistant to malaria naturally were the ones who were surviving versus the people that weren't. So for me, like, that's what I'm saying. It makes more sense to call it a symbiosis than a parasite, which is why I'm curious to have a conversation with Dr. Jackson just to see what she actually meant in terms of the parasitic relationship. Because I mean, the way that the term is right now is it means that the plant and human actually have a parasitic interaction, not the human with the mosquito, that would be the parasitic interaction. But again, like I said, I could be wrong, but I guess I'll just figure out a way to ask Dr. Jackson, because I would like some more clarification on that end.
Yeah, most definitely. You know, this is, you know, the information that we're discussing it's heavy information. So we might get a few different perspectives on how to approach it. But I think ultimately, some of the work that Dr. Jackson was presenting to us is for us to evolve our understanding about our relationship to the plants that are around us, as far as being able to sustain and increase our health, which is the reason why this presentation is happening. So that we can have a better understanding about how nutrition can be used as a way to prevent sickling within the sickle cell community, while also improving the health of our sickle cell community as well. So, you know, my suggestion is not necessarily to get hung up on the terminology, but to look at some of the outcomes of what our discussion was intended to do. Which was to broaden our scope on how nutrition can be used to eliminate some of the negative impacts that are being experienced within the sickle cell community.
Oh yeah, no, for sure. But the sad part about the reality of the world is, as soon as someone hears something, you know, they tend to kind of last on that one thing. And like I said, the way that you're describing everything, it just seems more of a symbiotic relationship. I mean, people in the scientific field, that's just kind of what we do. Was
that your first time hearing Dr. Fatima Jackson's presentation on plant parasite people coevolution?
No, that was my first time hearing plant human parasite evolution as being something positive and not being described as symbiotic. So that's why I'm like, that's interesting.
The video that we watched today with Dr. Fatima Jackson, was that the first time you watched that video?
Yeah, the first time I watched that video, but everything that she was talking about was making sense. But that's why I was curious why. And I don't think she mentioned, I mean, it just all sounded like symbiosis versus parasitic. That's the only thing I'm just curious about.
Well, I would, I would suggest that maybe watch it a couple more times, and then maybe things become more, clear to you why she came up with that concept and how she's using it. That was a video that, I had to kind of watch in bits and pieces and then kind of go back and watch a couple more times because she's a very intelligent woman, and the information that she is bringing to us is probably based off of maybe 40 years in the field. So some of that stuff that she's presenting on, we may not get it all at one time. So that's why that information is going to be available on this platform. You can come take a look at it anytime you want and watch it as many times as you want. And then once you have more clarity on it, then maybe we can discuss it. If you're not able to get in contact with Dr. Fatima Jackson, maybe myself and you can build on that concept a little bit more so we can work on how it could be more clear in the future so that we can better advocate for the sickle cell community.
Oh, yeah, for sure. That makes sense. Thank you.
Thank you. You may be on mute.
I'm sorry. Okay, yeah, I'm off now.
Okay.
You want to take that over there? Okay, I just wanted to say that this information was very invaluable and very useful and helpful. However, I think it needs to be a little more in layman terms and listening to her, because there was a lot of terminology that I didn't understand. And I'm sure I'm not the only one. So yeah, moving forward, maybe we could have further conversations where it can be broken down and simplified to some degree. But I really want to thank you, Christen, Mr. Brown, for your time and all of this great information and very useful information. And that's the ticket. We're just trying to be certain, SCAA is just trying to be certain that everybody gets all the information that is available out there, and not just from a clinical stance or a medical professional position, but from everyone that is living with sickle cell and doing their research on how to improve the situations, because we all know it's bad. So if there are ways that we can combat some of that negativity, it's great. And they don't have to go to the hospital and have to deal with people that don't understand. Then, that's what we want to bring aboard, people that can give out information that's helpful and useful for people living with sickle cell. And I have to say, you've done a great job today. And I appreciate everything that you've stated. And we would love to have you come out at our annual walk this year and have your stuff available for people to either have samples or just get that literature and that information